welcome everyone to this session called Mighty Molecules in Reproduction. So we're going to talk about biomarkers and their role in regulation in reproduction. We have a nice set of speakers and panelists today. So I encourage you, of course, to ask questions during the lectures or after the lectures. There will be a Q&A session at the end of the 3 lectures for today. But please enter your questions in the Q&A button at the bottom of the Zoom software. Do not use the chat for the question because someone is collecting the questions, and we will put them all together. Some of them will be probably answered during the talks, but we will go back to those questions for the Q&A. So please use the Q&A button. And so today, it's my pleasure to co-moderate this session with Dr. or Professor Marc-Antoine Driancourt. I have the pleasure to know Marc-Antoine for many, many years. So Marc-Antoine has a long career in animal science and animal reproduction. He worked in an academic setup at the INRA in France. And then after that, he switched to research industry. And he has worked for more than 35 years in that area. And last but not least, Marc-Antoine is the editor-in-chief of the journal, the scientific journal, Theriogenology, and he's going to tell us a little bit more about that. So, Marc-Antoine, the floor is yours.
Thanks, Pierre, for your kind introduction. I have the difficult task to introduce you as well. And honestly, I will keep it short because most of the crowd wouldn't know you very well. In a nutshell, what I want to say is that you graduated in France with DVM and a PhD. Then you jumped over the Atlantic and landed in Washington, where you still live and work. You are now Senior Program Officer for Science in the Office of the Undersecretary for Science and Research. And we team up together to keep Theriogenology going, and it's a pleasure to team up with you. We are also joined by Dr. Alison Bartolucci, who is an expert in morphology predictors of embryo potential. She recently published work generated during her PhD thesis on microRNA 21 in human cumulus cells, and she will act as a panel member. After saying that, I want to say a few brief words on Theriogenology. Theriogenology is an international journal dedicated to reproduction management, reproductive physiology, and reproductive biotechnologies. It is run by 3 editors, including Pierre and myself, and receives about 1,200 papers every year from all around the world. Amongst the main countries submitting papers, there are China, Brazil, and the US, which is not a big surprise because they are very large animal producing countries. The rejection rate of the journal is around 60%, and our readership includes mostly research scientists from all around the world working in the academic facilities. It also includes their master and PhD students, and also R&D staff from the animal health industry. Overall, this results in about 800,000 papers downloaded every year, which means that Theriogenology is a very respected journal. And finally, just a quick note to stress that 2022 started with a new launch related to Theriogenology. It's a companion journal called Theriogenology Wild, which is dedicated to wildlife with the same interests than Theriogenology. Theriogenology Wild is run by Fulvio Gandolfi, who many of you may know. Now, I think it's time to move to the real science. So I am happy to introduce the talk from Saadeldin. And so Islam is not going to be there because he's based in Korea. So in Korea, it's about 5 a.m. or it will be 5 a.m. when we finish the talks. So Islam will jump out of bed and join us. But now we are going to listen to his recorded talk. So welcome, Islam, and enjoy this presentation.
Yes, let me, before we start, just let me give a short introduction also to Dr. Saadeldin, because he has an interesting career. He has been extremely productive, even if he's a young investigator. He got his veterinary degree in Egypt, and then after that, he moved for a PhD to South Korea, and now he's still a postdoctoral fellow in South Korea. Interestingly enough, as I said, he's a very productive, and he received the Young Investigator Award recently from the International Embryo Technology Society. So this is going to be a very interesting lecture from him, and we will be pleased to be joined by him at the end of for the Q&A session.
Hello, everyone. My name is Islam Muhammad Saadeldin. I'm going to present the extracellular vesicles as non-invasive biomarkers for embryo quality. Currently, I'm in Seongnam National University in South Korea, and also I'm an associate professor at Zagazig University in Egypt. I have no conflict of interest to declare through this presentation and all the contents. First, I'd like to give a brief presentation or a brief information about the exosomes or extracellular vesicles. So exosomes and extracellular vesicles are bilipid-coated nanoparticles or nanovesicles secreted from most of the cells in the body. If you compare the size of the cells with the size of apoptotic bodies or microvesicles, and here is the exosomes, it's near or quite near to the size of the viruses. And here you can see the size by electron microscopy. It's very, very small if compared with the whole cells. But exosomes is not only the vesicles that we describe. So many extracellular vesicles are in this family. So like we have exosomes and microvesicles, it's quite bigger. And the matrix or calcifying vesicles, ectosomes, proteasomes, prostosome or oncosomes. So many vesicles are released from the cells and depending on the kind of the cells. But people are considering exosomes as a trash for the exocytosis or the release of the secreta from the cells. But exosomes are active bio vesicles, like it's a snapshot of the cells. If you can see here, this is the magnified part of the exosomes or extracellular vesicles. So each exosomes or extracellular vesicles contain much of bioactive material like microRNA, protein, and like mRNA, and even DNA. So this gives the exosomes the ability to be a good biomarker for many diseases, and also it can be used for the embryo quality monitoring. Mostly exosomes works as paracrine communication tool. So some cells secrete these exosomes and the bioactive materials inside the exosomes and released to the extracellular space or extracellular fluid. Then it can be picked up by another cell. So it communicates the paracrine communication through these bioactive materials. And there are some routine methods to how to isolate these EVs or extracellular vesicles and how to measure this concentration. Usually in our laboratory, or most of our laboratories use the nanoparticle tracking analysis. And in this part, if you have a conditioned medium or spin culture medium in the embryo culture, so you can see the size. The size ranges from 100 nanometer up to 500 nanometers, mostly around 100 nanometers. So these vesicles can be also tracked by the flow cytometry or nano flow cytometry, like this video you can see. You will see the motion or the Brownian motion of the extracellular vesicles, and it can be counted and measured in the concentration and in size through the nano flow cytometry apparatus like zeta view or nanoparticle tracking analysis. So let's give some presentation about my experiment in 2013. While I was a postdoc in Seoul National University, I was searching about the principle of feeder cells and feeder embryos. If we have like zona-free embryos and co-cultured with zona-intact embryos, and I found even this one in cloned embryos. So I found many blastocysts, maybe blastocysts reach over than 33% in cloned embryos. So I searched what is the principle about how the embryos communicate to each other. So I designed this experiment to make a co-culture parthenogenetic embryos and nuclear transfer or cloned embryos through the transwell dish or transwell implants. So we designed this experiment and we put the parthenogenetic embryo with the cloned embryos, and we cultured cloned embryos together in the same condition. So this idea given to me from Dr. Marius Ratajak. I attended a lecture for him in 2013 while I doing my experiment. So I learned what is the meaning of exosomes in this lecture. So also the concept how the exosomes can be released from the zona. If you can see this zona, so there is a bilateral communication or paracrine communication between the zona free and also the zona intact. So can exosomes pass this zona pellucida? So the answer was by Dr. Anne van Soom. She was very famous about how the viruses can enter the zona pellucida even for in vitro produced embryos. So I got this answer, and also I realized that exosomes can also enter or pass through the zona pellucida. So I first do the experiment in 2013 and published this paper by the first months of 2014, and it was the first report about the embryo-derived exosomes. So here we got the spent culture medium from the cloned and parthenogenetic embryos, and we examined through the transmission electron microscopy, and we found the exosomes around 100 nanometer or 130 nanometer. And surprisingly, we took these exosomes or extracellular vesicles and examined the content for mRNA. So we found this vesicle contain bioactive materials, especially mRNA for OCT4 and SOX2 and KLA4 and CMYC. These are known as the pluripotency factors. So the concept is parthenogenetic embryos can improve the cloned embryos through releasing these factors inside the microvesicles or exosomes, and it can be uptaken by the cloned embryo and increase or promote the growth of these cloned embryos. So we stain these vesicles through stain named BKH 67, and we visualize these nanovesicles inside the 4 cells and 8 cells embryos, and we found that these vesicles can be uptaken by the cloned embryos and improve this development. So after this research, I realized that this topic will be very important for embryology and the persons who are interested in embryo work. So I wrote a review article about what is the potential or what are the potentials implications of exosomes released from the embryo. And this review and my article in 2014 have been cited more than 200 times. So many researchers are interested in the embryo-derived exosomes and how the embryo communicate with the mother or maternal environment through these bioactive materials. If you can see, the endometrium can release exosomes and also embryo can release. So there are critical stage or critical period of embryonic maternal crosstalk and may be mediated by the exosomes or extracellular vesicles. So in this review, I asked 3 questions and the potential implications for these exosomes or embryo-derived exosomes. So I just give the hint to other researchers and also my research path. So I asked it first question about we can use this exosomes as a non-invasive method for judging the embryo quality and the implantation capability. And second, we can use these exosomes for sexing in case of male or female. And also it can be a prenatal genetic diagnosis method, or PGD, as a non-invasive method. So we have challenged how to use these exosomes. Very small amount of the medium, and very small amount of the bioactive material. So many researchers are doing this effort to clarify what is inside the message of these exosomes or extracellular vesicles. I completed my work with the collaboration of Seoul National University in Seoul and some other researchers in Saudi Arabia. So we investigated the effect of oviduct exosomes, and it can affect the dog oocyte maturation. Everybody knows that the dog releases the oocyte after ovulation. It's immature oocyte. So the maturation completes its duration, or M2 stage inside the fallopian tube. So what about the oviduct exosomes? We found that oviduct exosomes can affect or can improve the oocyte maturation in dogs. Also, we studied proteomics, and we found so many signals and proteins inside these exosomes. And also, I continued about the oviduct exosomes and its effect on the cloned embryo's development. We designed several sets of experiments how to use these exosomes through improvements, the parthenogenetic activation, or even the nuclear transfer, just co-culture with the medium, or injection of this exosomes solution in the perivital space in the M2 oocyte. And as we can see, the exosomes reduce the reactive oxygen species and oxidative stress, and also reduce the apoptosis and increase the lipid metabolism in these embryos. Then when I was in Saudi Arabia, so I studied what is the communication between male and female, especially in rabbits, because in rabbits, this model in rabbits experiment, we know that rabbit is an induced ovulator animal. So after coitus or mating with male, the ovulation occurs. So there are some signals inside the semen, especially the nerve growth factor. But we investigated if exosomes or extracellular vesicles can be also another factor or another mediator of this induction of ovulation. So we cultured the prostate and the testis and epididymis, and isolated the exosomes or extracellular vesicles from this primary culture. And we studied the effect on cumulus expansion and the first polar body extrusion and the meiosis. So we found that most of these mRNAs, especially the GnRH1 and kisspeptin and midkine and neurotrophin and nerve growth factor, several mRNA transcripts or signals are released inside the extracellular vesicles like prostate and testicle and epididymis. So we just assumed or speculate that there are some messages released from males and transferred through the exosomes or extracellular vesicles, and also can affect the female reproduction, especially in rabbit. And also this method can be applied for another species, especially camels and other induced ovulator, and maybe in human and other species. So let's go in depth. What are the current research in the in exosomes as a non-invasive method or biomarkers? We might know that many laboratories in human and other animal species use the morphology or morphokinetics or oxygen consumption or oxidative stress and several biochemical factors and proteomics and metabolomics as a non-invasive method for embryo selection. But what about the exosomes or extracellular vesicles? Also, I have to declare that just 3 days ago or 4 days ago, some report from Nature and Nature Medicine, they said that there are some problems with the PGD method, the genetic testing for the embryo. Is it feasible or not? Or is it mandatory to take the action or BGD for embryos before transfer or not? So we have to consider about the non-invasive method and to see what is the benefit of the non-invasive methods. So first, some researchers studied the genomic DNA contents and possibility to judge the BGD. And so many researchers said that the genomic DNA from the culture medium is a possible indicator for the developmental competence of human embryos and murine embryos, and it can be good for ART outcomes. And some researchers also said about the microRNA, and fortunately, Dr. Antonio Capalbo will talk about the microRNA in the spent culture medium, and he has a separate session about this one. But the microRNA is a good candidate for judging the quality of the embryos and how to judge the implantation or the sexual dimorphism, or even the developmental capacity, and even for embryo maternal recognition of pregnancy. And some other researchers look for the morphology and the concentration and just physical properties of these exosomes or extracellular vesicles. As you can see, some researchers see the concentration, some researchers see the diameter or the size of extracellular vesicles. So even last year, last 2 years, Dr. Fazili in UK, he cultured individual embryos and examined the spent culture medium from each or individual embryo and used as a non-invasive embryo quality markers for the bovine embryo. And I think it will be applied for human in the near future. So the major finding of the past studies, they compare the poor quality embryos and good quality embryos depending on the extracellular vesicle content. So mostly the good quality embryos has or have the EV size is less and EV concentration is less, while the poor quality embryos having the EV concentration is high and EV size is high. And also the DNA concentration in the spent culture medium is very high, while in the good quality embryos, the DNA concentration is less. But there are so many studies, but still, yet we want to validate the genomic DNA as a non-invasive biomarker for the BGT. Again, some researchers say that the apoptosis triggers also microRNA in the spent culture medium, and these microRNAs are released in the exosomes. So there are some factors like microRNA 294 is increased and significantly correlated with the apoptosis. So they can correlate the finding if you examine the microRNA 294 in the spent culture medium and you found high concentration of this or high level of this microRNA, it indicates the bad quality embryos. And even the oxygen tension and the oxidative stress on the embryos affect the release and the size and concentration of the EVs. If you can see here, the concentration of EVs are very increased in the high oxygen consumption. I mean the oxidative stress, while it's low in the day 3 embryos. Okay. And also, if you use white light and red light in your manipulation of the embryos, the white light is more toxic to the embryo. So in this experiment, they found the white light increased the concentration of EVs, and these EVs contains propedium iodide. It means genomic DNA inside these exosomes, and it will also affect the quality of embryos. So they consider that use red filtered light is advisable during the embryo implantation or manipulation in the IVF laboratories. And again, the same authors from the previous paper, they developed a sample and rapid flow cytometry method to identify the size and the genomic DNA content, especially for propedium iodide staining. And they found the concentration of the BI positive EVs are increased in case of implantation failure in case of human embryos. So this is very good indicator for the implantation failure if you use this simple and rapid flow cytometry through the staining of the EVs with propionamide. However, there are some recent reports said that even so many researchers said that microRNA and RNAs in the embryo culture medium, is it feasible or not? So until now, there is no gold standard microRNA to be reliable for sperm culture medium, and also deep sequencing results is contradicting to each other. So especially for tRNA fragment appears to be overexpressed in the conditioned IV medium and could be potentially novel biomarker for investigations. So until now, there are no consensus about which the microRNA or which mRNA could be the gold standard tool for the judging the embryo quality. And here also you can see the mRNA fragments in the in vitro culture medium also associated with the pre-implantation embryonic development. See here, there are two mRNA fragments like postin and VCNL. They are much increased in the pre-implantation development and correlated with the embryonic development in the exosomes or extracellular vesicles. And even the question for sexual dimorphism, there are some sexual dimorphism in the release of EVs, especially male and female. Particularly, if you can see here, the male and female MX expression for the ALEX and VBS4 and some mRNA transcripts are increased in case of male and reduced in case of female. So again, this is a relative expression between male and female. Again, now there are some sexual dimorphism between male and female, but here in case of the same study that we illustrated before, in case of microRNA 294, there was no sexual dimorphism between male and female in expression of this or releasing of this mRNA in the culture. In my experiment, I did this experiment in 2014 before leaving South Korea. I did a small trial to get the spin culture medium and analyze the Y-specific gene or SRY, and also this gene is X-linked or X-specific gene. So I found some contradictory results also, because we are measuring this expression in very minute amount of culture medium. I remember it was around 10 microliter. I cultured the embryos individually in 10 microliter culture medium. Then I took this culture medium and analyzed further the blastocyst for sexing male or female, and the embryo culture medium. I also analyzed the mRNA, but sometimes I cannot find the expression of GAPDH and the it's non-matching Y and X chromosome. So it needs much effort and much reliable data for this. Fortunately, I wrote a book in 2020, and I collected all the studies related to the embryo quality and how we can judge the embryo-derived extracellular vesicles. And you can see this book published in 2020 in Springer. So let's summarize what are the current situation about using exosomes or extracellular vesicles as a biomarker or non-invasive biomarkers for embryo quality. There are some limitations about using this method because there are some barriers about how to isolate the exosomes. We have many methods to isolate the exosomes, especially ultracentrifugation and the size-based methods like precipitation method, or affinity-based methods like special kit to get these exosomes from the spin culture medium. So every method has advantage and disadvantage, but there are some major barriers, especially EVs can be aggregated with the proteins, and it is non-specific for this culture. So in last year, I also edited an issue in Frontiers Veterinary Medicine, and we received a very nice review article wrote by Dr. Firaz, and they introduced the concept of microfluidics in the embryo culture medium and how to use the embryo culture medium through microfluidics for analyzing the extracellular vesicles and to isolate and characterize, and it can be as a potential for the embryo or standardized quality assessment. So they wrote that microfluidic method, it can analyze small device and also low contamination risk and rapid and easy for manipulation. And they have their own microfluidic devices and also micro mixer, and they can get on-chip micro mixer and embryonic EV isolation. So it is high-tech and promising technology for analyzing the extracellular vesicle released from the embryos through the microfluidics. So I will end my presentation about the challenges and the future direction and what is expected in the near future about exosomes and EVs research. The value and the market of the EVs is very promising. Currently, the market is around 46 million, and it's expected to be more than 10 times in 2027. So we need a reference and standard reference condition in the embryo culture medium, and this will be the good target for standardizing the embryo quality measurements. I'd like to thank my laboratory, Dr. Shaw and Dr. Lee and all the students. And also I'd like to thank Dr. Palmer and Dr. Comizzoli and Dr. Marc Antoine and all the organizers for the I3 initiative.
Hello, can you hear me?
Yeah.
Yes.
Back.
So I think we should all join together to thank Dr. Saadeldin for a very stimulating presentation. It raises to me obvious questions about stability and storage of exosomes for future use. And he will be present live later on today to address these questions and possible other questions that you may want to address to him. And before we move to the next talk, I would like to ask Alison whether she wants to comment on a few facts or findings of this talk. Alison?
Sure, thank you. Well, I found that to be a very interesting and intriguing talk, as I'm sure everybody did in the audience as well. What I found interesting was that in addition to potential use for biomarker analysis, there may even be opportunity for more research in terms of optimizing systems in our field, in the human, particularly in vitro maturation. I'm thinking about some of the work that he showed on the canines, the oocytes and the exosomes from the oviduct. He was able to improve maturation when the oocytes were exposed to that. And I think there could be potential applications in the human as well. If we look at extracellular vesicles and their contents from cumulus cells, perhaps there's an opportunity there. In addition, I thought the work on microRNA expression and exosome secretion in spent media when exposed to oxidative stress was interesting. And although it would be an expensive QC project, perhaps that could be used to optimize culture conditions as well, and in a more sensitive way to identify ways to optimize culture media. So
good. Thanks, Alison. So it's now time to move to the next talk. And with this next talk, we remain in the animal world because the next talk will be given by Eli Sellem, who has been working for the past 20 years on bull fertility. His final goal was to find a fertility predictor. He has focused on semen functionality tests and semen epigenetics such as DNA methylation, post-translational modification of protamine, and more specifically, the small non-coding RNAs. And this is what is going to present us in the next few minutes.
So, good morning, good afternoon, or good evening, depending where you are. I'm very happy to share this moment with all of you, and I would like to thank very warmly IVF Initiative for this invitation. So today we'll discuss about sperm-borne small non-coding RNAs and bull fertility prediction. First of all, some context elements to well understand the importance of the male fertility in our breeding industries. As you probably know, the genetic progress used the AI, or artificial insemination, as principal way of diffusion. Today, approximately 7 million of AI are realized in bovine per year in France. And of course, the use of less fertile bulls or less fertile ejaculates will decrease the global efficiency of the system and lead to important economical losses. Another very important point for this topic is that before the genomic selection, the qualification of the bull potential was done through the offspring's capacity, as for example, milk production. And several hundred of AI were done to produce these offsprings. And if the bull presented some difficulties in terms of fertility, the bull were rapidly identified and discarded or managed. And today, with the genomic selection, the genotyping, the bull potential is clearly identified early in the life of the bull, and the semen is disseminated very quickly on the field without any information about the bull fertility. That's why the identification of subfertile bulls or subfertile ejaculates, leading to their elimination before their dissemination of the field, remain a key concern for the breeding industries. And since many decades, different quality tests based on sperm functionality or sperm quality have been developed, as motility, viability, acrosome defect, morphological abnormalities, oxidation, mitopotential, DNA fragmentation, and so on, allow to clearly identify very particular bulls, but they are not allowed to predict the field fertility variation. And at maximum, when you combine all these tests, you can predict approx 40% of all the fertility, all the field fertility variation. So it's not enough to predict the fertility without any mistakes. That's why we need to find different kind of phenotype with the hope to increase our prediction strength. And that's why a few years ago, we decided to work with the small non-coding RNAs, to increase our prediction. A few words about small non-coding RNAs. The small non-coding RNAs belongs to the big family of RNAs. They can be coding or not coding, long or small, and etc. Today, we will focus only on small non-coding RNAs. They can be structurant or regulant. And you know two subfamilies of structurant small non-coding RNAs, the rRNAs and tRNAs, with a well-known role in terms of protein translation. But my favorite ones are regulant small non-coding RNAs, as microRNAs, piRNAs, rsRNAs, and tsRNAs, small fragments from the R and tRNAs. And these subfamilies play a very important role in the genome expression regulation via their action on messengers. So the most studied families of small non-coding RNAs are the microRNAs. They correspond to small fragments, approx 22 nucleotides. Half of them are inside genic region and mainly intronic. They can be alone on the genome or inside clusters. And most of them are transcribed by the RNA polymerase II in long fragments called pre-microRNAs. And these long fragments will be processed by different enzymes as DROCHA, dicer, exportin, and so on, to form a shorter fragment mature called microRNAs. And this fragment will be catched by a protein complex called RISC. And by this action, they can attach or catch messengers by complementary in terms of sequence. And this action will cleave specifically the messengers or block the translation. And in both of cases, it will lead to no protein obtained by this action. The tRNAs and rRNAs could be cleaved specifically by different enzymes, as angiogenin, dicer, RNase, and so on, to form shorter fragments called tsRNAs or rsRNAs. And these fragments have been discovered also inside the RISC complex, and they have exactly the same action on messenger than microRNAs. So no protein obtained. And finally, the last very interesting subfamilies of small non-killing RNAs, the piRNAs. For many years, piRNAs were considered to be specific to the germline, playing a role very important in the genome stability by fighting the retrotransposon elements. But a few years ago, several teams discovered that these piRNAs exist also inside somatic cells, and they discovered these piRNAs inside protein complex called here PI-RISC, and they have exactly the same action than microRNAs on the protein, no protein obtained. So you can imagine that with this kind of action, all the small non-killing RNAs play a very important role in all biological processes. And it's also true for the spermatogenesis. Several teams have highlighted this point by first discover a lot of different small non-killing RNAs inside the germline and germ cells. And they also highlight the fact that they are crucial for spermatogenesis by knockout mice for genes involved in microRNAs or piRNAs maturation. And these knockout mice lead to very important disturbance in terms of spermatogenesis, as described here, and in some cases, leading to sterility. Other teams have also highlight the specific role of some microRNAs, for example, the microRNase 34C, in the different stage of spermatogenesis. You can have all these reference here on the bottom of the slide. Okay, so you know probably that at the final stage of spermatogenesis, the spermatozoa are not functional at all. The epididymis journey is crucial for the final sperm maturation through the acquisition of several compounds. And it's through their interaction with extracellular vesicles called exosome or epipidymosome here that the sperm cells will acquire some specific compounds in terms of protein, lipids, glycans, but also small non-killing RNAs. And all these exosome or epidydymosome are produced by the epithelial cells of the epididymis. And it's very interesting to note that the content of these extracellular vesicles change according to the section of the epididymis. So the head of the epididymis, corpus, and coda. It's very troubling, this acquisition, because we suppose that the sperm cells at this stage are muted in terms of transcription. So what kind of biological processes these small non-killing RNAs will regulate? So it's tempting to think and to suppose or to imagine that these small non-killing RNAs will regulate some biological processes in embryo development. And a wonderful paper published in 2018 from Konin et al. Highlights exactly this fact in mice. They produce XE embryo with sperm cells located in the head of the epididymis or in the coda of epididymis. And they follow the developmental rate until blastocyst stages. And they found that according to the different conditions, no difference was observed in terms of developmental rates. But from the fourth cell stage to blastocyst stage, they observe a differential expression for 37 genes. They go one step ahead and they reimplant this embryo inside pseudogestant female, and they were not able to get birth with the embryo produced with sperm cells from the head of epididymis. Instead, the other condition, highlighting a very important point that the acquisition during the epididymis transit is very important for the embryo development after the blastocyst stage. So, to conclude this part, the small non-cleaning RNAs are required for a normal spermatogenesis. Some of them are acquired post-spermatogenesis. We hope that some of them are required during the first stage of embryo development, and potentially some of them shape the phenotype construction of the offspring. All these results was obtained in mice. And now what about in bovine? So to find a good predictor for field fertility, we need first to decipher two different points. First of all, the general catalog of ejaculated sperm in terms of small non-cleaning RNAs, and the dynamic acquisition of these sequences all along the male tract. 2 years ago, or 1 year ago, sorry, we obtained this kind of results. So here you have the relative expression of small non-cleaning RNAs in ejaculated bovine sperm. And as you can see, the sperm cells is rich in terms of small non-cleaning RNAs, but not in terms of diversity of the families, because only 4 families, so the microRNAs, piRNAs, rsRNAs, and tsRNAs, shape almost 90% of all the expression in small non-cleaning RNAs inside ejaculated sperm cells. The second interesting point is that a huge majority of these sequences are expressed in very low number of copies, and only 7% are expressed at more than 10,000 copies in sperm cells. And when we take a look on the dynamic acquisition of these sequences, the first observation was the kind of game of musical chairs among families and among localizations. So here, it's exactly the same kind of graphic. It represents the relative expression of small non-cleaning RNAs according to the localization, so the testis, the parenchyma testis, the head of epididymis, corpus, coda of epididymis, and 2 conditions of ejaculated sperm, so without seminal plasma and with seminal plasma. And as you can see, the sperm located inside the testis contain more than 80% of piRNAs. So the piRNAs are hegemonic in this localization. And as soon as the sperm leave the testis and enter inside the epididymis, the proportion of piRNAs decrease a lot to reach approximately 20% in ejaculated sperm cells. And the piRNAs are replaced by 2 other families, mainly the microRNAs here in yellow, and the fragment of tRNAs in orange, in blue, sorry. A strange observation was made in the sperm cells located in the corpus of epididymis, because here the main subfamily is fragment of rRNAs, but only in this location. And to be honest, we don't know why. Another way to explore this data is to plot the expression of all these small non-cleaning RNAs in a BCA graphic. And as you can see here, the different localization of small non-cleaning RNAs are clearly separated. And we have obtained a very interesting result that more than half of the global variability could be explained by the localization. And when we take a look on the differential expression according to the origin, you can see that the number of differential expressed small non-cleaning RNAs increase according to the tissular distance. So It's very interesting, all these results, but can we go through these results and try to find functional annotation? So for that, we focus only on microRNAs to find targeted genes of all these sequences. So the first step was to decipher the biological processes enriched by the microRNAs on sperm located in the testis. So the microRNAs represent only 3% of all the total microRNA expression. And as you can see here, two biological processes have been enriched and highlighted, ubiquitin and ERAD. And it's very interesting because these two processes linked to the organella and compound degradation are crucial during spermatogenesis. For example, knockout mice on genes targeted here by our microRNAs from the ubiquitin pathway lead to very important problem and disturbance in flagella and acrosome formation and lead to the sterility. So it makes sense to find this kind of targeting here. When we focus on microRNAs in sperm cell located inside the epididymis, they represent approximately the half of all the microRNAs expression. The target of these microRNAs are linked to the energetic metabolism. And different cake pathways have been highlighted as mTOR or AMP. And it's very interesting because, as you know, during the epididymis transit, the sperm cells acquire their motility. So it makes sense to find different pathways linked to the energetic metabolism highlighted here. And I advise you to read the publication of Silva et al. In 2019, because they explain very clearly the link between mTOR system and the motility of sperm cells. So once again, it makes sense to find this kind of genes highlighted here. And finally, when we take a look on microRNAs inside sperm located in post-epididymal tract, they represent approximately 40% of all the microRNA expression, and they highlight genes involved in tissular and embryonic morphogenesis, transcription regulation, heterochromatic organization, and the hypocake pathway was highlighted by these genes. So with in particular SMAD gene family. And it's also very interesting because this hypo pathway is involved in the stem cells renewal and the differentiation into ectoderm, mesoderm, and endoderm formation. So once again, it makes sense to find all this pathway highlighted here. So to conclude this part, the ejaculated sperm is rich in terms of small non-cleaning RNAs. Some of them are remnants from the spermatogenesis, and they are not totally or fully eliminated during the cytoplasm contraction. Other small non-cleaning RNAs are acquired via the extracellular vesicle all along the sperm journey inside the epididymis, but also in contact with the seminal plasma. And we hope that a part of these small non-cleaning RNAs could play a role in the first stage of embryo development. So, okay, all these results are very interesting and important to our point of view, but how we can transform this scientific knowledge into a real tool for our breeders. So we have tried to combine all these sequences to find a predictor of fertility. And for that, we have analyzed 154 bulls from two different breeds, Holstein and Montbéliard breed. We have chosen all these bulls according to their fertility value, and we have created two groups, the sub-fertile bulls and the sub-fertile bulls. And we have analyzed the small non-cleaning RNAs by RNA-seq with UMI technologies with 20 million tip reads. The first interesting results when we plot the expression of all the small non-cleaning RNAs we have found inside all these samples, that the first factors of differentiation is the breed. As you can see here in this BCA plot, here you have all the sample linked to the Holstein bulls and here to the Montbéliard breed. But the second axis, the y-axis, where we plot the fertility factor, we can clearly also separate the different group of fertility. The fertility explains only 3% of all the global variation among all these samples, but it's enough to find interesting potential biomarkers. That's why when we take a look on the differential expression analysis through a DSEQ2 algorithm, we were able to highlight approximately 600 small non-coding RNAs with a differentially expressed against or among these different groups. There are mainly fragments of rRNAs, iRNAs, and microRNAs. They are mainly underexpressed in subfertilized bulls. And to my point of view, the most important point is that a huge part of them are acquired after the spermatogenesis, so during the epididymis journey or in contact with seminal plasma. So highlighting maybe the fact that they could have really a good role and an important role in embryo development, and we hope so in the global fertility of the bone. Another very interesting point is that when we, all this a part of this microRNA, have been also and already studied by other studies published in human disorder in terms of fertility, as you can see here. So all these microRNAs are maybe involved in azoospermia, oligospermia, astenospermia, or idiopathic infertility, as explained by these different authors. Okay. The last point with all this data is that all the microRNAs are highlighted with the fertility comparison targets almost 40% of the most thousand expressed genes inside the oocyte, as STAT3, SMARCA5, SMAD family, BAS family, and so on. And they have also an important number of targets produced or expressed inside the embryo. And these things, this observation led us to trying to create our own hypothesis about the action of the sperm-borne small non-coding RNAs in embryo. As you know, the embryo needs to stay in totipotent stages for a certain amount of time. And the genes targeted here are all involved in the differentiation. So we made the hypothesis that the small non-coding RNAs bring by the sperm cells will play a role inside or play a role in this balance between pluripotency and differentiation. Okay, so last words about small non-coding RNAs in fertility prediction. We tried to go ahead with the black box predictors by combining the expression of highlighted small non-coding RNAs from the fertility comparison with an ensemble learning approach. So it's a mix between random forest and SVM algorithm. And we have done that only for Montbéliard breeds. And we have cut in two parts our population. 70% of the bull have been used to create this predictor, and we have tested this predictor on the rest of the population. And the first results we obtain are very interesting because almost between 80 to 90% of the sub-fertilized bull and the sub-fertilized bulls have been correctly identified and classified according to the fertility group. So, to conclude all these parts, some small non-coding RNAs seems to be linked to the male fertility, and these small non-coding RNAs are acquired at post-testis stages. So it's very interesting for us. And the combination of the small non-coding RNAs expression is possible with black box predictor, but it seems it seems, to give interesting results. We know that we have obtained these results with a low number of bulls. So the next stage is to increase the number of bulls in this population, and also to add bulls with normal value in terms of fertility, to avoid to have only 2 different classes and extreme classes of fertility. So I would like to warmly thank all my colleagues from Alice and INRAE, and all my colleagues inside the semen production centers involved in this project. I would also like to thank Apigen and ANR for the financial support, and of course, all of you to spend your time to be here today. So thank you a lot, and do not hesitate if you have any question. Thank you.
Okay, so that was a very, very dense and interesting presentation that really shows the value of studying, theriogenology and livestock species, really, because it's beautiful. And I like really to see the potential applications, but I know with this, the audience that we have today, probably people have in mind really what could be the potential applications to human IVF and human IRTs, especially for testicular biopsy, for example. Unfortunately, so Eli Sellem is not going to be with us today, but we have the possibility, we have recorded a couple of questions, so we may use that during the Q&A session later after the last lecture. So let me introduce the next and last speaker, last but not least, Dr. Antonio Capalbo he's the research from Italy, and he's the research director of eGenomics. And his research has been focused on the mechanisms of aneuploidies in oocytes and pre-implantation human embryos. He's been playing a lot of important role in the European Society for Human Reproduction and Endocrinology, especially the interest group in reproductive genetics. And is a member also of the Italian Society of Human Genetics, where he's leading a group of a working group on genomics. So, Antonio, the floor is yours.
Thank you. Thank you for the introduction, and welcome everybody. Let me also thank all the organizers of these and all the previous great events that are extremely exciting. And I'm very glad to be part of it for the second time now. Now, I have the task today of to take you now into the clinical IVF space and to review with you the evidence and our experience about profiling microRNAs in sperm blastocyst culture media in order to derive some kind of novel or to improve the current approaches that we have for embryo selection. So the first slide for me, this is a kind of disclosure about being a full-time employee in eGenomics. I strongly believe that any embryo selection strategy should start with assessment of the chromosomal constitution of embryos. And the experience that we have generated over the last 20 years is clearly telling that blastocyst biopsy is the gold standard today that we have in order to perform aneuploidy testing. And also in general, the working on cellular biopsies is for sure, on an analytical and technical perspective, for sure the best scenario that we may want to have as compared to liquid biopsies in terms of analytical validity. Now, we know that two-factorial biopsy is for sure, seems to be safe. We can consider it as a minimal invasive approach. It didn't show any effect on reproductive potential so far, nothing that is clinically relevant at least, no effect on prenatal or neonatal outcomes, and is in general accurate and reliable for aneuploidy testing. However, we also realized that this is not, this procedure is not being incorporated so far in all IVF laboratories, in all IVF clinics. And of course, this is suggesting that there are still some concerns concerning the equipment, the training costs, and for sure the addition of an extra procedure, which is in general not so complicated to be incorporated in the IVF workflow, but still can be considered a limitation. And if it's not properly done, this may cause some damage, or that will result in discarding or damaging perhaps viable embryos. So anything that can substitute this approach that is non-invasive, that is based on a very simple procedure such as collecting the sperm culture media, is of course welcome. Now, we heard already many things about non-coding RNAs or microRNAs, and the colleagues before, they have explained very well the biology surrounding microRNAs. And this is something that we start discovering as early as 1961, when the two Nobel laureates, Jacob and Monod, were the first describing the existence of structural and regulatory genes that today described as long non-coding RNAs, where microRNAs are the far more prevalent. These are RNAs, molecules that function as measured transcription and post-transcriptional regulators of gene expression. We know that today most of the transcripts of the human cells are regulated post-transcription by microRNAs. What are the important features of microRNAs for the context of non-invasive embryo selection strategies is for sure the fact that they've been detected in all human body fluids. There is a specific signature that we can underpin in all human body fluids, and this is suggesting that there is a specific biology footprint of microRNAs in each body fluid in our body. Other interesting evidence suggests that these extracellular microRNAs are extremely resistant, and they can be treated with extreme pH conditions, or they can be processed for several freezing and thawing procedures, and they seems to be extremely stable in the extracellular environment. So some of evidence that we have suggests also that microRNAs, they play a very important role for cell-to-cell communication, because as we saw before, they are stable extracellularly, and there is a specific microRNA aberrant expression in body fluids. And many efforts today and many new diagnostics applications are being developed by looking at microRNAs in several body fluids such as blood or saliva, and this is providing a lot of new avenue for developing diagnostic application for the early detection of several different chronic diseases such as cancer, cardiac, or immune-related diseases. So if we look at the mechanism of cell-to-cell communication, where there is a donor cell that synthesizes the microRNAs, release it into the extracellular environment, where they can go and reach the recipient cells, be internalized, and mediate the communication mechanism. Now, if we just try to change and substitute the cells with the embryo and endometrium, the hypothesis is sounding, where microRNAs can, of course, mediate an important role in the blastocyst endometrial dialogue aiming at implantation success. So the hypothesis that was behind our and other studies was that microRNAs can really be a significant, they can be significant contributors in the embryonic endometrial crosstalks, and by profiling microRNAs in the easily accessible sperm culture media, we may identify a signature which is associated with higher reproductive potential of human embryos. So in order to, I want just to show you our experience, we first tried to set up a protocol in order to profile microRNAs in such a, challenging samples that can be a sperm culture media that is collected after a few days of several cells growing in vitro. And at that time, we look and we choose a qPCR-based protocol, which is because this allowed very ultra-sensitive quantification of nucleic acids. And we actually look at a panel of around 1,000 microRNAs as a first screening strategy. So I think that it is always extremely important to validate properly any technology specific when we, in particular, when we look and we work in a small amount of samples, very challenging conditions, such as can be liquid biopsies and sperm blastocyst media, in order to get some data about the analytical validity of our protocol. We first compare bulk DNA, bulk microRNAs obtained from human embryonic stem cells, and we compare this from large samples versus small samples, showing that the correlation coefficient was similar between these two samples. And then we move to the analysis of the performance of this protocol for microRNA profiling on sperm blastocyst media. So here on the left side, you can see the first validation that we did was to evaluate the technical consistency in identical samples without biological variability. To do so, we have pooled 5 different samples of sperm blastocyst media. They were put in the same tube and then split again in 5 different tubes. In this way, we remove all the biological variation between the samples. And then we perform a blinded analysis with our protocol based on quantitative PCR. As you can see here, when removing the biological variability, the R value, the correlation coefficient was very high, on average was 0.94. On the other side of this figure, on the right side, you can see the analysis when the biological variation is kept. And here you can see that the correlation coefficient between samples, unrelated samples of sperm blastocyst media was 0.77, suggesting that the protocol was able to capture biological variability in sperm blastocyst media samples. Next, we further validated this protocol by looking at the correlation coefficient of twin embryos that were created by blastocell biopsy and from sperm blastocyst media collected from unrelated blastocysts. And as you can see here, also the protocol was able to distinguish between the twins, the sperm blastocyst media collected from twins and from controls. Next, we aim to characterize what was the signature of microRNAs that we can identify in human sperm blastocyst media samples, and to see and to check whether these microRNAs were of trophectoderm origin. And to do so, we've been analyzing the minimum signature of blastocysts and the relative sperm culture media. First, we characterize the presence and the signature of microRNAs in sperm culture media at different time points of human embryo development in vitro. You can see in this figure that it is very clear that embryos start to secrete microRNAs in the extracellular space soon after the compaction stage, while in the cleavage and the morular stage, we did not observe any difference in the concentration of microRNAs that can be detected. And this is suggesting that this active secretion mechanism of microRNAs begins with compaction or soon after the cavitation or at the cavitation stage. So to characterize the origin of microRNAs that we detected in sperm blastocyst media, we have compared and we have used, we have performed microRNAs profiling on human trophectoderm and ICM samples. To collect pure ICM and trophectoderm samples, we use a micromanipulation and mechanical procedure of ICM isolation that we developed back in 2011 when I was working with Dr. Neji in Atlanta. We developed this method to isolate pure ICM samples from human blastocysts that was extremely reliable and effective. And we applied this method here in order to isolate ICM trophectoderm samples that were then profiled for the microRNAs. And here you have a short summary of these results, where you can see that on average, we detect 59 different microRNAs in sperm blastocyst media. 97% of them were also expressed from the trophectoderm, and only 2 microRNAs were more abundant in the sperm blastocyst media as compared with trophectoderm. So this evidence suggests that there is a specific packaging and exporting mechanism for the delivery of extracellular microRNAs that is primarily coming from the trophectoderm. At that point, then we wanted to see if any microRNA can be related with implantation, and if it was a specific signature which is able to distinguish or to predict embryos that are going to plant or those that are going to fail implantation or to result in miscarriage. And at this early stage, we run a preliminary analysis where we pull 28 non-implanted sperm blastocyst media coming from non-implanted blastocysts and 25 sperm blastocyst media coming from embryos that result in an implantation or in a live birth. And we have performed a preliminary comparison on these clinical outcomes. This is a summary of the results. You can see this is a volcano plot where on the y-axis you have the adjusted value, while on the x-axis you have the differential gene expression between implanted and non-implanted embryos. Only 2 microRNAs showed to be significantly different between the 2 groups of embryos, and these microRNAs were also tested for functional analysis, and they were shown to be important for endometrial cell proliferation. So with this information on our ends, we decided to validate a clinical model on a larger multi-center study where we aim to analyze single euploid embryo transfer cycles in different clinics, so with different embryo culture conditions, as well as with different culture media and IVF settings. So the clinical results are described in this paper. Just going straight to the point, in order to make this assay compatible with clinical use in terms of cost and turnaround time, we have designed a specific straightforward plate qPCR approach that was including 30 different assays for 30 different microRNAs that were run in triplicate, plus exogenous controls, negative controls, of course. And this plate was designed to analyze 4 different sperm blastocyst media samples, and the turnaround time is compatible also with fresh embryo transfer because it's about 3 hours from sample collection to results, and also the methodology was already designed to be cost effective because, if I'm not wrong, it was something like 20 to 30 euro per sample. And this design allowed us to scale this approach to a larger sample size, of course. So here you have the results of our clinical validation. You can see that in the right side, you can see that we run 229 and 21 samples, sperm blastocyst media samples. Approximately half of them result in a live birth, and the others fail to implant or result in a miscarriage. And by looking at the p-values or the fold change, you can see there are some significant difference in the concentration of microRNAs implanted versus non-implanted embryos. However, it's important to know that when we incorporate all this information in a multivariate logistic regression analysis, including also blastocyst morphology, as a covariate, you can see here that still some microRNAs remain significant at the p-value level, but the odds ratio is, as you can see here, very close to the unit 1, meaning to say that the statistical effect of this predictor is very weak. A final consideration that I wanted to make is that once we have ruled out and upload this in our embryo selection strategy, we need really to put into the context what can be the clinical utility of adding additional biomarker that may improve embryo selection beyond euploidy. We know that there are several categories of patients. The ones that are advanced maternal age, they are producing very few euploid embryos, 1 or 2 embryos. So we really need to think about how can additional biomarkers change or improve the clinical practice once we know that plantation rate of euploid embryos is pretty much consistent, resulting in a live birth in approximately half of the attempt. And where you have 1 or 2 embryos, I mean, improving selection, I'm just thinking to what extent this can be relevant for the clinical practice. On the other hand, when we have more euploid embryos to choose from, we have seen this publication using a very large data set from the RMA New Jersey that after 3 embryo transfer of euploid embryos, you can get something kind of cumulative live birth rate, which is higher than 93–94%. This is just to stimulate the discussion at the end and also to give you some food for thought. Unless these non-invasive biomarkers are also able to replace invasive aneuploid assessment and provide also additional features of the embryonic genetics that we do not assess by aneuploid testing. I'm really thinking what can be the role of or the place of new biomarkers for improving embryo selection on the top of euploid. This is something that, of course, we can discuss at the end. Now, take-home message is that the population of microRNAs secreted by human blastocysts in sperm culture media has been characterized in our works as well as in the study of others for the first time in IVF. We have seen that human embryos start to secrete microRNAs at the time of compaction, and that microRNAs that we find in the sperm blastocyst media are mostly derived from trophectoderm or from trophectoderm and ICM cells. Validation data for key microRNAs on large data set of euploid embryo transfer failed to improve or to improve the practice or the prediction upon implantation. And of course, in our work, I need to acknowledge that we focus our analysis of at the beginning on something around 300–400 microRNAs. So there is still room for checking and testing other microRNAs, as well as we have today new technologies also to use and to try to expand our knowledge about microRNAs in sperm blastocyst media, and that may also provide a more reliable assessment with lower technical variability. From basic research perspectives, as we also heard in the previous presentation, outlining the roles of microRNAs during linear specification and blastocyst stage, and on implantation may, of course, help improving the clinical embryology. And of course, I need to acknowledge also this was the work of many colleagues, and that are all displayed here, the principal IVF sites, as well as the academic institution that will be collaborating for this project. And also I need to acknowledge that this project was also supported by the GFI grant from Mercer. Thank you all very much for your attention, and of course, I'd be happy to take any question you may have.
Thanks a lot, Antonio, for a very exciting presentation. I may want to start with one double question to you, and then I will leave the stage to Pierre and Alison for further questions. So my question is, firstly, you picked up 3 microRNAs that you could relate to the birth of a live baby. And your conclusion was that they were good, but inadequate for a good prediction. So the first question is, if you combine the 3, would you improve prediction to a level where it would become meaningful? And the second question is, you looked specifically at microRNAs, but Eli Sellem in the previous talk looked and found quite interesting findings with the piRNAs. So are you aware of the detection of piRNAs in your spent blastocyst media? And if so, is that promising to explore this route as well?
Thanks for both the questions. So regarding the first question about the prediction model, all the three microRNAs, yes, we're trying to build up predictive models combining the three microRNAs that still were still significant at the p-value level. But when you put into the equation, you put morphology, then all the effects of these predictors become extremely weak and not meaningful for clinical practice. Regarding the second question, indeed, there is a lot of work still to be done in this field. We started working on this subject many years ago. At the time, qPCR for microRNAs was the best technology in our hands to explore this field. Now, today, of course, we have more reliable and comprehensive methodology in order to characterize the whole population of long non-coding RNAs that we may find in extracellular, the embryonic extracellular environment. So this is something of course, for future investigation, it is worth pursuing.
Good. So Pierre and Alison, the stage is yours.
Hey, so yeah, let's open the Q&A. Thank you so much, Antonio. And let's open the Q&A session. We have a lot of questions to the different speakers, but first of all, I can tell you that we have received also a lot of congratulatory messages for the three speakers. And so thank you so much. And welcome, Professor Saadeldin. It's good to see you.
Hello. Good
Thank you
afternoon.
for joining us. Well,
Yes,
since
thank you so much.
you made this huge effort really to wake up early for us, let's start. Let's start with you with some questions for you.
Yeah, sure.
Okay, so we had let's start with a very simple question from Philip Chan. Can you hear me, by the way?
Yes, I can hear you.
Okay.
Yeah.
So a question from Philip Chan. He would like to know how stable are the exosomes and do they degrade rapidly, either by spontaneously or by enzymatic degradation?
That's a good question. Usually, extracellular vesicles are lipid vesicles, so it can resist the trypsin or botulitic enzymes, so it cannot be degenerated easily. But there are some research that if you put the exosomes in the normal temperature or ambient temperature, so the contents can be somewhat changed because there are some mRNA or microRNA, so it can be generated within the time. So, but it can be stable if you freeze these exosomes in -80 or liquid nitrogen, so it can be preserved for a very long time.
Okay. Thank you so much for that first question. Alison, do you have a question for any of the speakers right now?
I did have a question actually for Dr. Capalbo. So this is a loaded question, I understand. So if you had, let's say, 2 euploid embryos that were morphologically equivalent but had differential expression of mRNAs that you had, identified as having a correlation with implantation, what do you think is could be the root cause of that? Is it something that happened in the oocyte? Is it something that happened in the environment during culture? What would your thoughts be on causes for differential microRNA expression?
Well, this is very speculative, but let me try it. So, what can be the cause? The cause can be something in the programming of the embryos, in how is able to deliver, to establish a good communication with the surrounding endometrium. So probably what I can expect is that the correct expression of microRNAs and secretion of microRNAs in the extracellular environment is aiming to prepare and to stimulate the endometrium to have a better receptivity at the implantation site. And this can be due to several events happening during culture. So embryos can be a little bit late in this process, or but still, I mean, this is something that need to be properly investigated. We still have no functional data about mechanism of the release of these microRNAs into the extracellular environment. We are trying to tackle this issue by looking at the clinical potential application, trying to derive also basic biological insights by looking at the clinical value of these microRNAs that we could profile in the extracellular environment. But as I show you, I mean, we fail to detect meaningful differences between euploid, reproductively competent and non-competent embryos.
Thank you.
Okay, so go ahead, Alison, if you have other questions.
Well, I was just going to pull one from the audience here. Let's see. Well, Dr. Sellem's not here, so let's do another question for Professor Islam. Do you know, this is from Pascal Salvetti, do you know what is the minimal duration of individual culture period of embryos to have enough biomarkers in the culture medium? And is there a consensus on that point?
Also, this is a good question. Usually, we follow up the embryos or the cultured embryo for several days. So some said about 2 days or 3 days according to the duration or the stages of the embryo. So if you monitor the embryo in 8-cell stage or 4-cell stage or morula or blastocyst, so it depends on the stage of the embryo, what kind of the changes inside the embryo stage. But I tried this in 2013 and 14, so I can see mRNA expression from 4-cell stage and morula and blastocyst. So at any stage you can see the exosomes or extracellular vesicles.
By the way, something related technique question, but the media that you use for in vitro culture medium, for in vitro culture of embryos, are you sure that they are depleted from any extracellular vesicles?
Yes, that's very, very good question, because we use a chemically defined medium. We avoided the BSA or any source of animal protein. We replaced with some metabolites, and we are sure that by electron microscopy we cannot find these vesicles if we compare this embryo cultured and the plain culture medium or without embryos. So we already confirmed that the chemically defined medium doesn't contain the extracellular vesicles.
Excellent. Okay, I know that Eli is not with us, unfortunately, but Marianne, could you, I mean, we had the opportunity to record 2 questions and 2 answers from Eli. So could we play that? If you're not ready, okay, perfect. You're ready right now. Thank you so much. So the question was, what is the relevance of these small RNA molecules?
microRNAs could be very interesting because they seems to share something very relevant in terms of embryo development in several species, as in human. So I think this one could be very interesting biomarkers for field fertility. If we take attention on the results obtained by Conin and al. I suppose that it will maybe have some problems because the sperm cell is not fully mature, and the acquisition during the epididymis journey was not here. So it could be dangerous is not the right term, but I don't know how we can obtain a good product if we done these kind of things. But it could be different between mice and human, of course. But yeah, to my point of view, all the acquisition after the spermatogenesis are very important and required for a normal embryo development. So we need to take into account of all these things. That's why, for example, for bovine, we think about the development of a new kind of media containing all the relevant small non-coding RNAs present inside exosome in aiming to increase the results in terms of fertility after animal artificial insemination. We can maybe think about the same kind of techniques or approach in human, maybe.
Okay, other question was microRNA molecules are often quoted as being unstable.
It's the hypothesis by a lot of scientists, but to be honest, no. It's the fact that these RNAs are very short, make them very strong, and it's difficult to eliminate them. But inside the oocyte, and after a few hours or days inside the embryo, we don't know how many times the small non-coding RNAs bring by the sperm cells will stay there, and what kind of impact they can have really in the embryo development. But you know, they can have two different impacts, in fact, and direct impact on the regulation of messengers already present inside the oocyte and shape maybe the first stages of embryo development, but they can also have an indirect impact by leading some DNA methylation because they can recruit some enzyme to bring methylation on DNA and then modify the genome expression in long range. So maybe after the blastocyst stage and after that also. So I'm sure that even if they are not present in the oocyte or embryo several days or several weeks and so on, they can have impacts several days after the fecundation or several weeks after the fecundation.
Okay, I'm glad we got the opportunity at least to get something from Eli. So let's continue with Antonio and Islam. Alison, do you have more questions for Dr. Capalbo?
Let's see what the audience has here. This is from Bronte Stone. In addition to determination of blastocyst quality by morphology, what marginal gain and likelihood of implantation is provided for the SCM analysis?
Well, the point is that really what I was trying to also to convey as a message is that we really have to discover very powerful biomarker of implantation, right? Because after we have excluded aneuploid embryos from our cohort of available embryos in IVF cycles, I mean, we have not so many choices to make. So usually patients of advanced maternal age they are producing very few euploid embryos. And in general, what we see in many studies, and this is a consistent findings that we see in different practice worldwide, is that apart from morphology, which is bringing a slightly improvement in the prediction beyond euploidy, all the other parameters usually are not so relevant or significant. That's why this was the point of one of my last slides in the presentation, that we really need to think about what can be the clinical utility of developing new biomarkers for embryo selection that are bringing small improvements for prediction upon implantation. Of course, there are many contexts, many realities in the world where embryo biopsy is not feasible because this is, forbidden, or because many are still not convinced about clinical utility of the GTA. So in those contexts, developing some biomarker that can improve prediction upon implantation, that for sure maybe it can be valuable and meaningful for clinical practice. But when we compare it with aneuploidy testing, and then we add on the top morphology, is challenging now to find out some really powerful biomarker that can significantly improve the practice on the top of this. At least this is my experience and my reading of it.
Good. Excellent. Thank you. Okay. Let me ask questions to Professor Islam. Terry Morgan has a question and says that most scientists working on extracellular vesicles do not think that DNA or a fragment of DNA or messenger RNAs are in exosomes, in small extracellular vesicles. Do you think that there is definitely messenger RNA and or DNA in larger microvesicles? What is your opinion about that?
Yes, that's a very good question, and recently answered by some papers that small extracellular vesicles didn't contain the DNA, genomic DNA, so but contain some messenger RNA. So it depends on what is the kind of these vesicles. As I mentioned in my presentation, these vesicles range from exosomes, microvesicles, apoptotic bodies, and so many other forms of vesicles. But genomic DNA are not exist in the small extracellular vesicles. It means less than 100 nanometer or 120 nanometer. But messenger RNA can be found in these kind of vesicles, like microvesicles and exosomes. Yes.
Okay. Alison, can I ask a follow-up question?
Yeah, sure.
Okay. So another question for Islam, actually. It's not necessarily follow-up, but Konstantin Boyarsky is asking, what do you think about the presence of extracellular vesicles from human endometrium during the peri-implantation period of the embryo?
Wow, this is my current experiment in porcine. So I am now isolating the endometrial-derived exosomes or extracellular vesicles and co-culture with a new or novel approach. I'm using the like matrigel or extracellular matrix for the embryo attachment, in contrary to the feeder cells. So I'm using just matrigel or extracellular matrix to see the interaction and what is the effect of the endometrial EVs on the blastocyst stage day 7 or day 8 from porcine species. So it's very interesting to study this, particularly in human. And there are many researchers, they are doing this experiment for what is the effect of the endometrial EVs on the embryo in pre-implantation and even at the implantation stage. So it's exciting area of research.
Yes, definitely. Thank you. Thank you. Alison?
Well, I think maybe one more. This is again for Islam. I think it may be a question that a lot of people are wondering, especially given the audience of human embryologists. How would we translate this technology? And I guess this could be, open to anyone. How would we translate this technology to everyday use in the IVF lab? This was a question from Michael Reed. What equipment is readily available, and are there any manufacturers that are exploring this for the bench use in the IVF lab?
Yes, that's very good for the practical use. Usually if you are looking for embryo quality checkup or monitoring, so you can culture the single embryo culture, and you have to optimize the single embryo culture, and within 15 or 20 microliter or more or less, according to your optimization. And you can take the spent culture medium, just a few amount or a few samples from this spent culture medium, and you analyze the target of your study, of your marker. So if you are studying metabolites, or if you are studying mRNA or microRNA, so a small amount of the culture medium is also challenging for the analysis. So first you have to optimize what is the reliable method to detect this small amount of DNA or microRNA or messenger RNA. And also I saw in the question there one method now, it's nanofluorescence flow cytometry. Recently I used this method for checkup of the small quantity of exosomes or extracellular vesicles, and the device machine is named ZetaView. ZetaView, it's like fluorescent nano flow cytometry. So it can also monitor the size and the concentration of exosomes for just checkup of the embryo quality.
Thank you.
Actually, there is a question for you, Alison, from Marc-Antoine.
To broaden the discussion, Alison, I tend to remember that you did studies relating microRNAs and cumulus cell function. Would you be so kind to say a word about your findings?
Sure. So it was for my PhD thesis. I focused on one particular microRNA, microRNA 21. And so I looked at I stripped the eggs individually and then analyzed the expression of this microRNA and compared it to that egg's ability to make a blastocyst, as well as the aneuploidy result, or the PGT result. There was no correlation with the euploidy, but there was, as you might expect, with the egg's ability to make a blastocyst. So the higher levels of miR21 5P, the more likely it was to make a blastocyst. So, I mean, that was sort of just the initial, and I would like to continue that work, looking at lots of contributing factors from the cumulus cell into the oocyte, particularly as it relates to in vitro maturation.
And I have a further question for Professor Islam. Coming back to exosomes, is that doable to freeze them? And if so, what is the right procedure?
Yeah, that's great. So usually we isolate the exosomes or extracellular vesicles in PPS solution. So we can freeze in -80 or liquid nitrogen. But there are some researchers raise a question about how can we add like cryoprotectants on this medium? So because after freezing and thawing, the membrane can be ruptured. So they are looking now for the optimum cryoprotectant solution, and with minimal effect on the content of extracellular vesicles. That's very good. Yes.
Thank you.
And actually, I just had a comment, because we've been talking about microRNA, small non-coding RNA, piRNA, and all those nice mighty molecules that are part of the exosomes or extracellular vesicles. But we have studied in our group also the presence of proteins and other factors also that are enclosed in those extracellular vesicles. And what's been very interesting also to see is that especially for epididymosomes during the epididymal transit, there is a lot of conserved also proteins across different species in those extracellular vesicles. So evolution has definitely kept some of those content of extracellular vesicles during the epididymal transit, of course, mostly in mammals, but that's also something that we need to keep in mind, because it's not only the microRNA. And by the way, oh, sorry, Alison, you wanted to ask a question?
Oh, no, I think sorry.
Okay, no, I had actually, regarding the microRNA, there was a question for you, Antonio, because the question, let me make sure that, a question about from Krishna Pavani. Do you think circulating microRNAs could be potential biomarkers, because they are easily degradable so as soon as they are released into the extracellular matrix space? But what do you think about that?
Circulating microRNAs in the extracellular environment again?
Well, circulating, my understanding is probably circulating in the blood of the mother, right? I mean, is that a possible biomarker?
Well, yeah, I mean, why not? Something we've seen that many diagnostics are being developed based on that, right? So why not to investigate something that is related with infertility or with other outcomes or reproductive outcomes? As long as I know there is nothing being done in the human, or nothing that has been consistently reported and that show reproducibility in different study, but for sure something that is interesting for further investigation.
Okay, thank you. Thank you, Antoine. Alison, Marc-Antoine, do you have any other questions, or do you see more questions from the audience?
Not from my side.
Okay. We are approaching anyway. I think we've been together almost for 2 hours, which is nice. I think we can definitely thank again our speakers. So Antonio, is late for you at night. Islam is early in the morning. For Eli, it would have been also late in the evening. For me, it's right in the middle of the afternoon. So I could, and for Alison the same. So we could stay, online for a longer time. But we're going to let you either go to bed or start your day. And thank you again. A round of applause to the speakers for today. It's fantastic. We had a lot of, and thank you for the people who attended from all over the world. Also, we had several hundreds of participants today, so thank you so much. So in terms of, to close, I will let Marc-Antoine say the final word, but to close, I would like to extend also our thanks to everybody who took part of the webinar. So my co-moderator, of course, Professor Marc-Antoine and Driancourt and Alison Bartolucci, but also our speakers, as I said, Islam, Eli, and Antonio. And of course, we don't want to forget the team from the International IVF Initiative and the organizing committee, including Jacques Cohen, Peter Nagy, Shaista Saadeldin, Marianne Cetevez, Giles Palmer, and Thomas Elliott, and of course Victoria Almela, who was behind the scene capturing all the questions and putting them, in order. So thank you very much, everyone. So Marc-Antoine, I will let you for the final words.
Thank you, Pierre. So I would like to extend my thanks to all of you who listened for 2 hours. Please make sure that you join again soon the International IVF Initiative meetings. The next one is scheduled on April 19th and is called More or Less. So please look at the internet site to learn more about it. Also, feel free to look at Theriogenology. As you may have realized today, there is a lot of useful information on animal science in it. So feel free to use it for your publications when you work with animals. And that said, I wish you the best for the day. For the people from down under and from Islam's side, have a great day. For Antonio, have a good meal now. It's time probably for a late meal for you. And for all of us, thank you for the good science tonight.
Thank you so much.
Thank you, everyone.